Healthy human biology requires individual cells to follow a finite lifespan. Programmed cellular death is a vital function that maintains the balance of the entire system. This lifespan is governed by a physical countdown clock located at the extreme ends of our chromosomes, known as telomeres. The telomeres consist of a repeating genetic sequence, TTAGGG. Every time a cell replicates, that sequence physically shortens. When it completely runs out, the clock hits zero. The cell reaches its limit and enters programmed self-destruction. A cancer cell ignores this biological countdown entirely. It continues dividing in an infinite loop, bypassing the body's standard expiration date. The deadliest weapon a cancer cell possesses is its capacity to operate outside these biological restrictions, achieving immortality by circumventing the body's internal security systems. Inside a rogue cancer cell, the replication cycle proceeds differently than in healthy tissue. The cell begins its survival strategy by activating a specialized enzyme called telomerase. During division, telomerase rebuilds the shortening ends of the chromosomes, adding new genetic blocks to replace the lost sequence. This rebuilding freezes the countdown clock. No matter how many times the cell replicates, the telomerase length remains constant. The cell replication cycle is a strictly regulated process, featuring distinct phases for growth, DNA synthesis, and actual division. This flowchart shows specific security checkpoints. Before a cell moves through the G1S or G2M junctions, it must pass inspection for genetic errors. These are monitored by the P53 tumor suppressor gene. If damaged, P53 locks the checkpoint, preventing further replication. To bypass this inspection, the cancer cell mutates to disable the P53 gene, effectively removing the guards from the checkpoints. With P53 neutralized, the cell can push severely damaged genetic material through the replication cycle without triggering any alarms or forced cell death. Even after bypassing the main checkpoints, the cell still faces an internal maintenance crew known as the mismatch repair or MMR system. Normally, MMR genes scan, ensuring perfect AT and CG pairing, fixing chemical typos they identify. As the DNA sips together, the cancer cell disables the MMR genes. This allows a chemical typo, like an A nucleotide binding to a G, to slip past the repair crew without being corrected. These unchecked genetic errors multiply rapidly with every single cell division, creating a massive, aggressive biological momentum that cannot be stopped. By disabling this final layer of genetic repair, the cell transitions from an isolated anomaly into a compounding mass that no longer recognizes its own internal errors. These integrated survival mechanisms allow cancer to persist and expand despite the body's standard defenses. Because the disease utilizes our own biological machinery, it operates with the same efficiency as a healthy cell. However, the mechanisms cancer uses to survive also expose its specific vulnerabilities. Targeted therapies are now designed to exploit these pathways. Researchers can design compounds that inhibit telomerase or leverage broken MMR genes to overload the cancer cell with errors until it can no longer function. By identifying exactly how cancer cells bypass the rules of mortality, medicine can reactivate these internal alarms, forcing the cell to face its own end.